Hello, my name is Randal Burks. I'm a junior biology major from Waldorf, Maryland. Today I will present my study on the modification of the Thayer-Martin medium to effectively select 424 commensal and pathogenic Neisseria species. My research collaborator was Gabrielle Scales, and our research advisor was Dr. Goitia from the biology department here at Spelman College, and she guided us throughout our fall and spring semesters of the 2022 to 2023 academic year. There is a crucial role of the gonococcal isolate surveillance project, also known as the GIST project, where they monitor the susceptibility trends of Neisseria gonorrhea strains and guide the selection of gonococcal therapies in the United States. GIST aims to monitor trends, characterize phenotypes of isolates, describe resistance, and the investigation of male patients with urethral gonorrhea. Within the background, in the United States, Neisseria gonorrhea is the second most commonly sexually transmitted infection, and its resistance to antibiotics is increasing. The Centers for Disease Control and Prevention, also known as CDC, has classified gonorrhea as an urgent threat due to its resistance to multiple antibiotics. And furthermore, commensal Neisseria can transfer antibiotic resistance to pathogenic Neisseria through horizontal gene transfer, which makes it really difficult to control the spread of antibiotic resistance in Neisseria gonorrhea. During my research, I aim to answer the question of how to modify the GIST growth medium to promote the growth of commensal Neisseria species specifically. During my research, I conducted a disc diffusion assay as the first experiment. This assay measures the susceptibility of microorganisms to different antibiotics by examining the diameter of the zone of inhibition around the antibiotic-soaked discs on GCP agar plates. To test the antimicrobial agents, I use 19 commensal Neisseria bacterial strains, which includes nystatin, colistin, vancomycin, trimethoprim, and SXT. By measuring the zone of inhibition using the ImageGauge software, I determined the commensal Neisseria strains were if they were susceptible or resistant to the antibiotics. If the zone of inhibition ranged between 0.6 or 0 to 6 millimeters, it indicated that bacterial resistance was occurring to the towards the antibiotic. An image on the screen to the left displays the measured zone of inhibition and how to determine it. The presented graph depicts the average diameter of the zone of inhibition of the antibiotic colistin versus the respective commensal ID number. Among the 19 commensal Neisseria strains subjected to testing, 2 displayed resistance to colistin with an average zone of inhibition of 0. In contrast, 17 of the tested strains exhibited susceptibility to colistin. These results indicate that the colistin is a promising antibiotic for further testing in the context of combating Neisseria bacterial infections. SXT is a combination of trimethoprim and sulfamethoxazole, which we employed as our initial choice of antibiotic due to its inclusion of trimethoprim, which was the only antibiotic available in our laboratory at the time containing this ingredient. The accompanying graph exhibits the average diameter of the zone of inhibition in relation to the commensal ID number. The results reveal that out of the tested commensal Neisseria strains, 18 were susceptible to SXT. To comprehensively evaluate the impact of trimethoprim, we procured trimethoprim for further experimentation on these commensal Neisseria strains. Upon experimentation with trimethoprim, I discovered that all 19 commensal Neisseria bacteria exhibited susceptibility to the antibiotic. These findings were strongly suggesting that trimethoprim would be an appropriate candidate for further testing in context of combating Neisseria bacterial infections. The outcomes obtained from the susceptibility test conducted on commensal Neisseria bacteria were as expected, as nystatin is a drug primarily used for antifungal purposes. While both vancomycin and nystatin were not displayed in this presentation, they exhibited similar results with resistance towards the antibiotic with all the commensal Neisseria. These results for nystatin revealed that all 19 commensal Neisseria bacteria strains were resistant to nystatin, indicating that this drug is not effective against this type of bacteria. Moreover, the susceptibility tests also showed that 18 out of the 19 commensal Neisseria strains were resistant to vancomycin, which is also known as a broad spectrum antibiotic used to treat bacterial infections caused by gram-positive bacteria. This result is concerning as vancomycin is becoming increasingly common in clinical settings. And Also, based on these findings, it is recommended that further testing be carried out on Neisseria bacteria strains using colistin and trimethoprim antibiotics, as these drugs have shown effectiveness against similar bacterial strains in previous studies. So, colistin is a polypeptide. For background and understanding of what colistin is. It's a polypeptide antibiotic used to treat infections caused by gram-negative bacteria, while trimethoprim is a synthetic antibiotic used to treat bacterial infections caused by gram-positive and gram-negative bacteria. So, in conclusion, the susceptibility test conducted on commensal Neisseria bacteria revealed that the expected resistance to nystatin and concerning resistance to vancomycin. So furthermore, or further testing with colistin and trimethoprim antibiotics is recommended to determine the effective treatment options for Neisseria bacterial infections. So the following experiment that I conducted was minimum inhibitory concentration, and as I proceed throughout this presentation, I'll refer to it as MIC. So the purpose of this presentation, or the purpose of this experiment, as I meant to say, is the determination of the minimum inhibitory concentration at which trimethoprim and colistin can inhibit the visible growth of commensal and pathogenic Neisseria species. So I tested 23 commensal Neisseria species and one pathogenic Neisseria species, which was also known as the NGO, or Neisseria gonorrhea, by diluting each isolate using a 96 well plate and pipetting 5 microliter drops using the multi-channel pipette. I plated these samples on a gonococcal base GCB agar solid medium with increasing concentrations of trimethoprim and colistin to assess the each of the isolates' resistance. To the right of this text, I have presented an illustrative example of the 96 well plate, which includes 4 distinct sections, is what I did during my experimentation. And in each section, it consisted of 6 different isolates. So for each 4 sections, 6 isolates went in, equaling to the 24 isolates that I used during this experiment, and each of them were diluted. So additionally to the accompanying image showcases the spots that were generated on the GCB plates, which is to the right of the 96 well plate. So based on my testing using the disc diffusion assay, I have selected colistin and trimethoprim as the antibiotics to use for the MIC experiment. So the graphs displayed on the screen for the MIC of each antibiotic against the Neisseria sample being tested. So sample 43 and through 66 consists of commensal Neisseria, while sample 67 is the pathogenic Neisseria gonorrhea. So on the graph for colistin, 17 commensal Neisseria samples have a minimum inhibitory concentration within a similar lower range, while the 7 other commensal Neisseria exhibit varying ranges of MICs. So the Neisseria gonorrhea has an MIC of 11.25 micrograms per milliliter. From these results, it can be concluded that the recommended MIC for colistin for most commensal Neisseria would be within the range of 17 the 17 samples displayed, averaging of a 0.8 micrograms per milliliter. Regarding trimethoprim, the majority of the commensal Neisseria samples fall within the range of 20 micrograms per milliliter, suggesting that this will be the recommended minimum inhibitory concentration for trimethoprim. So according to the results of the MIC experiments, it is recommended that the gonococcal isolate surveillance project include a concentration of 0.8 micrograms per milliliter for colistin and 20 micrograms per milliliter for trimethoprim in their surveillance program for commensal Neisseria. So next steps that we would potentially develop Next steps that I would potentially develop for myself for a new project would be new growth medium recipe by incorporating antimicrobial concentrations based on the data obtained from DDA and MIC experiments. The modified media will then be validated through the spot testing to confirm the selection of commensal Neisseria. Once the media is confirmed to be effective, we will finalize the modifications to the protocol and share the results and potential publications with CDC for review and possible publication. By sharing the findings, we hope to contribute to the understanding of antimicrobial resistance in commensal Neisseria and aid to the development of better antimicrobial therapies. So as I conclude, I would like to present my citations for the presentation. And thank you each for listening to me discuss my research. And I really hope you enjoyed my presentation on the modification of the modified Thayer-Martin medium to select for 24 commensal Neisseria and pathogenic Neisseria species.